Hi everyone, this is Lance Capel and this is my signal transduction presentation. I decided to do my presentation on receptor tyrosine kinases, and they are a class of cell surface receptors, and they are activated by binding to their specific ligands, such as growth factors. So a little bit of background on what receptors are. They are very important for cell signaling. Receptor tyrosine kinase is a receptor that present on cell membrane. They act like antennas, which then receive signals from outside and then perform activities inside the cells. The molecule that binds with the receptor is called the ligand. So why are receptor tyrosine kinases important? Well, they're important for performing normal cell function. So when a ligand binds with its receptor, tyrosine kinase, it phosphorylases, that means add phosphate on its specific site. This will trigger signaling inside the cell. And they also help in maintaining cell growth and development of tissues. The receptor tyrosine kinases trigger a cascade of intracellular signaling pathways. And these pathways regulate various cellular processes, including proliferation, differentiation, migration, and survival. In cancers, receptor tyrosine kinases can become dysregulated, leading to uncontrolled cell growth and tumor formation. This dysregulation can occur through various mechanisms such as overexpression of the receptors, mutations that cause constitutive activation, or autocrine, paracrine loops where cancer cells produce their own growth factors to stimulate RTKs. So now let's discuss receptor tyrosine kinase and cancer. So the role of tyrosine kinase in cancer, when over signaling of tyrosine kinase occurs, uncontrollable growth of cells starts. RTK's hyperactivation may lead to suppress apoptotic genes that may reduce the death rate of a cell. This will lead to adhesion properties and movement of cells called metastasis. So if you see here on this chart, they transport adenosine triphosphate to substrates which have remnants of tyrosine. They are found on the surface of cells. They also exist on transmembranes as receptors. And they are protein in nature, and they aid in the movement of cells to block proliferation of cancer cells. Some more information about their role in cancer. So they participate in growth and the division of normal cells. They can absorb nutrients through pathways, which is used in cancer therapy. And they also kill tumor cells through apoptosis. Also, excess amounts of RTK can cause proliferation of tumors. So now let's talk about the types of receptor tyrosine kinase. First is vascular endothelial growth factor. Next is the platelet-derived growth factor, epidermal growth factor receptors, and then we have the fibroblast growth factor. So now let's get into the kinases that play a role in cancer. We have the EGFR, the HER2, the ALK, the VEGFR, the MET, the non-receptor tyrosine kinase, the BCR-ABL, and then we have the SRC. So the epidermal growth factor receptor, the mutations or overexpression are common in lung, colorectal, and head and neck cancers. The human epidermal growth factor receptor 2 is overexpressed in a subset of breast cancers, leading to aggressive tumor growth. The anaplastic lymphoma kinase, this is where gene fusions like the EML4, ALK are found in subset of NSCLC. And then the vascular endothelial growth factor receptor. This promotes angiogenesis, which is the formation of new blood vessels, which is crucial for tumor growth. The mesenchymal epithelial transition factor. Mutations and amplifications are seen in various cancers, including lung, liver, and kidney cancers. The BCR-ABL, which is a fusion protein resulting from the Philadelphia chromosome translocation in CML, leading to unregulated kinase activity and cancer progression. The SRC is a non-receptor tyrosine kinase that, when overactive, contributes to various cancers by promoting cell proliferation and survival. Next, we'll dive into the tyrosine kinase inhibitors in cancer treatment and the mechanism of action. So some examples of TKIs is the name Glivec, which targets BCR, ABL, and CML, leading to high remission rates. We have the Tarceva, which targets EGFR and NSCLC, which is effective in patients with specific EGFR mutations. Then we have the Herceptin. This targets HER2 and HER2 positive breast cancer, improving survival rates. Tyrosine kinase inhibitors are small molecules that specifically block the kinase activity of RTKs by binding to the ATP binding site in the kinase domain. And this prevents phosphorylation and activation of the receptor and its downstream signaling pathways, inhibiting cancer cell growth and survival. Next, tyrosine kinase inhibitors can be highly effective, particularly in cancers driven by specific RTK mutations. However, resistance can develop due to secondary mutations in the kinase domain that prevent TKI bonding. Activation of alternative signaling pathways that bypass the inhibited RTK. So let's talk about some side effects. Some common side effects include skin rashes, diarrhea, fatigue, which are generally more manageable compared to traditional chemotherapy. Some tyrosine kinase inhibitors may cause more severe side effects, necessitating careful monitoring. Okay, let's talk about the platelet-derived growth factor receptor. The platelet-derived growth factor receptor has two receptors, which are alpha and beta kines, and is made up of 4 ligands. They are present in the testes, the brains, and the kidneys. They're also comprised of some stem cell growth factor. When uncontrolled, aids in sprouting and splitting of tumor cells. It presents 2 pathways, the mitogen activated protein kinase and the PI3K AKT in therapy. And then next we will talk about the vascular endothelial growth factor receptor. This is manufactured by cells that work together to produce blood vessels. It is a type of platelet-derived growth factor which is made up of cysteine. It results from them being overexpressed in blood and other vascular tissues. It is important in cancer research and studies due to its vascular permeability structure, and they are also harmful to the patient. Next, we'll discuss the fibroblast growth factor receptor. So, breast cancer is a devastating cancer in women. Worldwide, it is the first in the list of terms of frequency of all tumors, and it is second only to lung cancer in terms of mortality. Briefly, fibroblast growth factor receptors are receptor tyrosine kinase members encompassing the cellular membrane in a single region, and therefore considered single-pass membrane proteins. They are made up of N-terminal extracellular domains with 3 different immunoglobulin-like subdomains, which are D1, D2, and D3. A transmembrane domain constituted of an alpha helix and an intracellular region, which has tyrosine kinase motifs capable of phosphorylation and a carboxyl terminal end. So, as we discussed, the fibroblast growth factor receptor are important in curing breast cancer. This is caused by the mutation on the gene lines. And again, this type of cancer is very common in women of reproductive age. These code for the fibroblast growth factor, and in the presence of a defect in the body, they can also become cancerous. And lastly, they are processed by macrophages. Next is the epidermal growth factor receptor. This causes cell growth. Some of them include cetuximab, panitumumab. I mispronounced those. They contains a transmembrane which uses its enzymes to complete the roles played by receptor tyrosine kinase. It has monoclonal antibodies which are used in pharmaceutical manufacturing of cancer drugs, and it is found on the upper surface of the skin. As far as epidermal growth factor receptor stimulation, this acts by binding to EGF receptor on the cell surface and stimulating the intrinsic protein tyrosine kinase activity of the receptor. And it also initiates a signal transduction cascade. As a result of this, a variety of biochemical changes take place within the cell, including increased intracellular calcium levels, glycolysis, and protein synthesis and transcription of certain genes, which has ultimately led to DNA synthesis and cell proliferation. So in general, binding of its ligand, epidermal growth factor, results in stimulation of the EGFR tyrosine kinase, which in turn stimulates intracellular signal transduction, enhances transcription of growth-related genes, and it also promotes cell growth. So how are receptor tyrosine kinases used in cancer treatment? Well, the receptor tyrosine kinase receptors are used in multiples to prevent drug resistance when used singly. It can be toxic to normal cells. Multiple pathway response is the most recommended. It uses 2 different strategies, and it's vertical and horizontal pathway inhibitions. The major mechanism is through apoptosis by the use of concanavalin A, con A. So this could result from blocking the first conjugate, Horizontal pathway inhibition prevents the triggering of a second carcinogenic pathway. The vertical pathway inhibition attacks the same tumor twice. So this topic I find to be very interesting, fascinating. This is the cancer drugs made from the receptor tyrosine kinase. So this is used for lung and gastrointestinal cancer treatment. The sunitinib term Sutent, they're used in glioma and esophageal cancer therapies, and is common among oncologists for colorectal and lung cancer. And the bevacizumab, which is also known as Avastin. And not to get too off course, but one of the reasons why I chose this topic is because I deal with Avastin. I work for a retina specialist clinic, and I work with ophthalmologists, and the eye doctors use Avastin to treat several eye and retinal diseases like age-related macular degeneration. AMD is a leading cause of blindness in people over 50 years of age in the U.S. And basically it's caused by the breakdown of the central portion of the retina, which is called the macula. And that's a very sensitive part of the back of the eye. Avastin also treats diabetic macular edema and central retinal vein occlusion. And the macula is basically responsible for the detailed central vision in the eye. I just wanted to throw that out there since I deal with Avastin, and I also found out before working on this project that it is also used for cancer therapy, which ties into what I'm discussing today. Examples of drugs from RTK and targeted cancers. It works against colorectal cancer. The imatinib is also called Gleevec. It relieves chronic myeloid leukemia and gastrointestinal metastasized tissues. They have been proven to be successful against mammary carcinoma. The patanimab, which is commercially known as Vectibix. The lapatinib, referred to as the trastuzumab, and Tykerb, which I probably mispronounced. The imatinib is a tyrosine kinase inhibitor, which is used in the treatment of Philadelphia chromosome positive chronic myeloid leukemia or metastatic malignant gastrointestinal stromal tumors. In normal cells, tyrosine kinase enzymes are turned on and off as required. In CML cells, one tyrosine kinase enzyme, the BCR-ABL, is always on. The imatinib blocks this BCR-ABL enzyme. As a result, these cells stop dividing. Because the BCR-ABL tyrosine kinase enzyme exists only in cancer cells and not healthy cells, imatinib works as a form of targeted therapy. For example, only cancer cells are killed. And the Vectibix is a therapy that blocks growth signals from reaching the cancer. Receptors on the cell surface receive signals instructing them to grow and divide. So just a recap of what I discussed in this presentation. For the receptor tyrosine kinase, they are essential for various cellular processes, including survival, migration, differentiation, and also cell growth. Receptor tyrosine kinase is present on cell membrane, which specifically binds with ligand and perform action by phosphorylation and dephosphorylation of their specific site. When over-signaling of tyrosine kinase occurs, uncontrollable growth of cells starts. Receptor tyrosine kinase's hyperactivization may lead to suppressed apoptotic genes, and that may also reduce the death rate of a cell. And this will lead to adhesion properties and movement of cells called metastasis. Also, the receptor tyrosine kinase are involved in several signaling pathways that regulate fundamental cellular functions. Dysregulation of the receptor tyrosine kinases has been implicated in various diseases, highlighting their importance in maintaining cellular homeostasis. Again, the TKIs have revolutionized the treatment of cancers with specific genetic mutations, offering targeted and more effective therapy options. Ongoing research aims to overcome resistance mechanisms and develop new TKIs. TKI's broader application in cancer treatment. And these are my references for my presentation. I hope you enjoyed listening to me, and I hope you enjoyed my presentation. I hope you found it informative, and thank you.